how's it going? So now we're going to talk about our T helper cells and our adaptive immunity. So our T helper cell is going to be a type of T cell, and this cell is going to be very important. So we have our T helper cell. Now this cell is going to have a protein known as a CD4 protein. And I'll talk about what that is in a little bit. So we have the CD4 protein. And if you remember from what we talked about with the macrophage earlier as an antigen-presenting cell, we're going to have that over here. Now our antigen-presenting cell does not only have to be a macrophage, it can be a B cell that has recognized this antigen and now produced the MHC2 complex. It can be a macrophage like we talked about, or it can also be a dendritic cell. And so the main thing about the antigen-presenting cell is that it's going to have this epitope, or this bacterial peptide, located on the outside of its cell. And I'll draw that in red. And so the importance of that is for the T helper cell to now recognize, is this cell one of us, or is it a foreign cell that we don't want inside our body? And so our T helper cell is going to have this T cell receptor here, which is going to be able to recognize that. Is this one of our cells, or is this a cell that is foreign to us? And so if this cell is foreign to us, we're going to use this CD4 protein that I talked about. And the CD4 protein is going to kind of act like a hook. And it's going to latch on into this antigen-presenting cell. And now we're going to release these communication molecules known as cytokines. And so these cytokines are going to tell this antigen-presenting cell to now kill itself and die. But the cytokines also have a different function as well. And these cytokines are going to be released, and I'll draw it over here. And these cytokines are going to tell our naive B lymphocytes, naive B cells, to now produce two different types of B cells. And these B cells are going to be very important in fighting our infection that we have. So our naive B cell is going to turn into a plasma B cell, and it's also going to turn into a memory B cell. Okay, so our plasma B cell is going to be very important in fighting this infection as quickly as possible. So this plasma B cell is going to secrete antibodies. And these antibodies are going to have different varying functions on how to kill this infection here that we have. So now these antibodies are going to go out into the bloodstream, they're going to go out into the mucosal membranes, and they're going to kill this infection that we have. However, we're also going to produce these memory B cells, and these memory B cells are going to be clones of this antigen here. And so basically what they're going to do is they're going to produce a bunch of cells that will just stay in our immune system for the rest of our life. And what that's going to do is it's going to basically, if we ever get this infection again, we're going to be able to release these memory B cells. It's going to be a lot quicker, and we're going to have a lot quicker fight against this infection that we might get again in our lifetime. So the T helper cell is very important in terms of being the boss cell. It's going to recognize whether this antigen-presenting cell, whether this epitope is one of us or whether it's foreign. And from that point, it's going to release these cytokines, which are communication molecules, and those molecules are going to stimulate the production of our plasma B cells as well as our memory B cells. All right, so now we're going to talk about the T killer cell. This cell is going to be another type of T cell that we have in our bodies, but it's going to have a little different function as compared to our T helper cell. So our T killer cell, first let's draw, so let's imagine this as a somatic cell, or any different cell in our body. And so we know that our cells are going to have a nucleus, they're going to have an endoplasmic reticulum, And so let's say that this somatic cell gets infected. So let's say we have a virus here. So this virus is going to come in, it's going to release the virus into the cytoplasm of our somatic cell. Well, what this cell is going to do is it's going to have this thing known as an endosome. So this endosome is going to take in this virus here, and it's going to produce these little epitopes, or little amino acids, of our virus. And so our ER, or our endoplasmic reticulum, is going to produce a little something different, and this is going to be known as an MHC1 complex. And so the main difference between our MHC1 complex and our MHC2 complex is that our MHC1 complex can be on any somatic cell in our body, whereas our MHC2 complex is going to be specific for our antigen-presenting cells that we talked a little bit about earlier. So this MHC1 protein is going to load these little epitopes onto its little site here. It's going to come out onto the outside of the cell, and it's going to load this epitope on the outside of its cell. And so now the somatic cell has this epitope on the outside, and we know that this cell, so we know that this cell is infected. So where does the T killer cell come in? Well, our T killer cell is going to have a very important protein known as a CD8 protein. And so the T killer cell is going to have to do two different things before it can kill this somatic cell. The first thing is it's going to have a T cell receptor, and it's going to have to recognize whether or not this cell or this epitope is foreign to us. If this epitope is foreign to us, we're going to kill the somatic cell. If it's not, then we're not going to. The other thing is that the T killer cell is going to have to receive the signal from our CD4 cells. So if you remember, our CD4 cells are T helper cells. So it's going to receive the signal from the T helper cells, and that's going to release cytokines. And now our T killer cell is going to be able to kill this somatic cell. But let's end this video with just basically compartmentalizing our adaptive immunity. So we know our adaptive immunity is going to break off into two different types. We have our B cells, and we have our T cells. So our B cells, we're going to produce both plasma B cells as well as memory B cells. Now our plasma B cells are going to secrete antibody to fight that infection. And the plasma B cells are going to help with the first time we get this infection. So it's going to be our primary immune response. And this can usually take 7 to 10 days, which isn't that quick. Now our memory B cells are going to produce clones, and these clones are going to be saved in our body for the next time we get this infection. So these are going to be responsible for our secondary immune response. And so the next time we get this infection, we're going to secrete a bunch of these memory cells, which are going to be able to take care of that infection a lot quicker. So this can take hours to days, which is a lot quicker. So now let's talk about our T cells. Two important ones that we talked about in this video are going to be our T helper cells and our T killer cells. So our T helper cells are going to be basically the boss cells of our immune system. They're going to be the ones that are going to release the signal to produce these B cells, but they're also going to release signals to tell our T killer cell to kill its cells. So one important thing with the T helper cells is that they're going to recognize our MHC2 complexes. And they're also going to have CD4 proteins. Now our T killer cells, they're going to receive the signal from our T helper, but they also have to recognize that epitope on our infected cell. So it's only going to recognize cells that have the MHC1 complex. And so the T killer cell is going to have a CD8 protein. T killer cell is also going to release perforin, whereas our T helper cell is going to release cytokines. So that's the immune system for you. I broke it down into our B cells and T cells here, but hopefully this video helps.